So this is Noah and Colin's process mitosis. What you're seeing now is spermatogenesis. The chromatids are finding their sister chromatids and making pairs. And after that, they will move towards the center and find their homologous chromosomes. And this is a diploid cell, and right there they crossed over and just separated GNA and shared GNA, shared genetic material. That was them pulling apart. So now the cell is splitting, and you have two new cells with each having the 46 chromosomes. Now you will see both cells are splitting again. This time they're pulling the chromatids apart and they're splitting once again. So each cell has some genetic material from the mother and some genetic material from the father. And these form the gamete sperm. Now this is oogenesis. Once again, you have your genetic material matching up, moving towards the center and crossing over. This is just to create more variation in the species. The centrioles are forming, the spindle fibers are moving towards. What you don't see is the nucleus breaking down. The chromosomes are pulled apart to opposite sides, but however, this time what occurs is the cell splits in a new, different way. So this is different from mitosis. Otherwise, it's very similar. It splits into one small and one large cell, and the small cell goes away and the big cell stays. Now the chromosomes are being pulled apart into two separate chromatids, and so the new cell will have parts from the mother and parts from the father, but they'll all share the same DNA. So then the sperm meets the egg cell and forms a zygote, and a human life is formed. And now this is the sped up version. That was spermatogenesis, and this is oogenesis. And now for the next thing, it's just a video showing.
The cell prepares for meiosis just as it does for mitosis. But as the DNA condenses, each chromosome finds its homologous counterpart. These matching pairs of chromosomes hold equivalent genetic information, one set from mother and one from father. Next comes the first shuffle of nature's genetic deck. The two homologs trade genes in a process called crossing over. This forms new hybrid chromosomes. The spindles then place maternal and paternal chromosomes randomly on each side of midline. It is another shuffle that can lead to a host of different genetic outcomes. Finally, spindle fibers pull the homologs apart. This leaves the sister chromatids intact. With twice the amount of genetic information the cells will need, nature must cut the genetic deck in two. This second division produces 4 unique sets of chromosomes. Because they carry half the genetic information, we call these haploid cells. Many of them will mature into sperm and eggs, the reproductive cells called gametes.